Thank you for having me here. Today I'll be talking about the applications of analytical techniques to cosmetics. In today's talk, I'll be discussing the utilization of different types of analytical techniques to the cosmetic industry. And I'll use some real-life examples to show you how our Jordi Labs to use these techniques to answer the why question. So a brief discussion on who we are. Jordi Labs was founded in 1980s and soon becomes a leader in the plastic analysis. And we accomplish over 1,000 jobs each year. And the objectives of this job can range from, extractable and leachable analysis to quality control testing to investigations. And thankful to the cutting-edge instrumentation we have here at Jordi Labs and our very talented chemists, we are able to serving a wide range of industries, including pharmaceuticals, medical device, food and beverage, and of course, cosmetics. The objectives of cosmetic analysis really depends on what our customer is seeking for. But in general, the goals can be classified into the following categories. First, we could conduct the formulations of a certain product and give you a comprehensive understanding of the chemical constituents of this product. We can also do quality control testing to help our customer to have a better quality control on their product. Next, we can do some purity testing, and for example, the purity testing can be applied to answer some failure questions and figure out why and how to solve these problems. And next is a very specific area in cosmetic analysis. It's pigment testing, which we can also do here in-house. And last but not the least, it's the regulatory analysis to comply the regulations. Given the complexity of the cosmetic products, we pick the different techniques to tackle different types of compounds which has different molecular weight, polarities, and volatility, etc. For example, LCMS is used to analyze the compounds which is polar and ionizable, while GCMS is used to analyze the volatile and semi-volatile compounds. While GCMS and LCMS are used mainly for the organic compound analysis, we can also use ACME-DEX and ICP-MS to analyze the elemental compositions of a product. Another thing we can do here is the FTIR, which can give you the bulk chemistry of the product. Later on, I'm going to discuss the utilization of each of these techniques to certain projects and show you how these techniques can be used to solve certain problems. The first example I'm going to show you is a deformulation of a soap bar. You would imagine that the chemical constituents of a bar soap should be pretty simple. It's a soap. However, it could be more complicated than you think. So thankful for the cutting-edge techniques we have at Jordi Labs, we were able to give a comprehensive analysis on the chemical constituents of the bar soap. So the technique we pick here include LCMS, GCMS, ACME-DEX, and the DSC. As I mentioned earlier, LCMS is used to analyze the ionizable and polar compounds, such as detergents. And HGCMS is particularly good for the detection of volatile compounds, such as fragrance. So this technique is very powerful for cosmetic analysis because fragrance and aroma compounds is widely applied in the cosmetic products. Another advantage of HGCMS is the sensitivity of this technique. It can pick up signal at a PPT level, that's parts per trillion. This is very important because the for some of the fragrance compounds, they can significantly affect the smell of your product, even at a trace level. So we really need a method which can have a low detection limit. Another one we use is the EDEX, which can give you the information on the elemental compositions, while DSC can give you information on the overall structure of the product. So let's move to the first technique, the LC-MS. And for this particular job, we picked Q12 LC-MS. The advantage of this technique is that it has an ultra high sensitivity and accuracy on the mass. So it's very good for quantitative analysis because it actually can have a mass accuracy to 5 places decimal points. After we acquire the LC-MS data, our scientists will use the data and compare this with the commercially available libraries. We also heavily rely on our Jordi library to do structure elucidations. And sometimes we also use LC-MS fragmentation patterns to help us to identify the unknown compounds. So the next slides show the MS data we obtained from the Q12 LC-MS. As you can see, we compare the chromatogram of our product with the blank, and we identify each of those unique peaks which represents to one unique compound. And this really gives us the comprehensive information on all the ionizable and Porter compounds in the formula. And we can see we got quite a lot of identifications on the chemical constituents of this certain product. And by looking into the chemistry of these compounds, we can see that there are some non-chain fatty acids, which is not surprising because it's a soap. And we also found sulfonic acid. Sulfonic acid or sulfonate is applied to the product to help lather, because sulfonic acid can lather better compared to soap. It does not create the insoluble substance in the hot waters. That's why the sulfonic is used for this product, because it can lather better than the soap. After we used LC-MS to do the qualitative analysis, we then moved to the quantitative analysis. The detector we use here at Jordi Labs include UV and CAD. So UV is good for the compounds which has chromophores, while CAD is a more general detector which can give you quantitation values on the compounds which does not contain chromophore. And there are different types of quantitations we do here at Jordi Labs. As you can see, the difference is really the accuracy of these methods. So for example, for formal quantitations, it has the highest accuracy because we compare the signal of our analyte of interest with the standards, which is the analyte itself. So it has the highest accuracy. We also do relative quantitations, which is a slightly lower accuracy, where we used internal standards to generate the calibration curve. And semi-quantitation has the lowest accuracy, but you can quantify a very good numbers of compounds in one single analysis. So what we do here, for example, as you can show, as showed in the slide, we integrate the peak area of each unique compound, and we compare the peak area with the standards we used, and then give a quantitation value. All right, so after LC-MS, where we detected detergent as ionizable Porter compounds, we moved to GC-MS. In this study, we picked HGC-MS because it's a very clean and fast method to detect volatile compounds. What we can see here is the major volatile constituents in the subbar is different types of terpenes, and this could explain the citrus smell we obtained from this product. Next, we move to ACM index, which can give you the elemental information on this product. In this particular project, we use index to confirm our findings from the LCMS and GCMS. So as you can see, the index analysis showed us the presence of carbon, oxygen, sodium, sulfur, and chloride, which totally makes sense because, for example, sodium chloride is added to the fatty acids to give you the salts to make soap. And sulfur is a good indication of the sulfonates we observed from the LCMS data. DSC is a technique which can give you some thermal information of the product. For example, it can give you information such as melting point, glass transitions, and thermal transitions. In this case, we ran DSC and we also observed two distinguished thermal transitions, which is an indication that this product is a mixture of compounds rather than one pure compound, which again confirmed with what we found earlier. So a brief summary of the deformulation of the bar soap. We used different techniques including LCMS, GCMS, ACM index, and DSC to tackle different types of compounds. The LCMS data showed us that there's a non-chain fatty acids and some surfactants and sulfonates in the product. And the HGCMS showed us the major volatile components in the bar soap are terpenes. And then ACM index and DSC kind of confirmed our finding from the LCMS and GCMS data. Some other analysis we could do, including ICP-MS and IEC, which can give you quantitation values for soluble ions and elements. We can also do FTIR, which can give you the information of the bulk chemistry of your product. And I'm going to discuss some of these techniques later. And the next project I'd like to discuss is a very fun project. It's a deformulation of rose. So everyone likes rose, but we keep asking us what makes rose smell like rose. So we did this project on Valentine's Day, where we took some fresh rose and we did headspace GCMS. As I mentioned earlier, headspace HGCMS is a very powerful technique to give you structure information on the volatile compounds. And another advantage of this technique is that it does not require sample prep, so you can actually put a rose petal as is to the chamber and then you get your data. So what we saw from the analysis is a mixture of different fragrance compounds. So the cocktail of the compounds actually make rose smell very unique. And another thing we observe is that the most abundant component in the rose petal is this 3,5-dimethyltoluene, which is widely applied in the cosmetic industry. Another thing I want to mention is that this compound is unique to this species of rose, which is a Chinese rose, and it's absent in other type of rose like European rose. And this compound gives the Chinese rose the very special tea flavor, which you do not have for other rose types. Another thing is that DMT is applied to many cosmetic products because it has the sedative smell and can help you relax. So if you go to some aromatherapy, sometimes they will use this compound to help you relax. So earlier I talked about how we use different techniques to deformulate the certain products. And another aspect of deformulation is that we can use these techniques to compare different products. For example, we can compare the brand names with generics using these techniques. So one day we got an inquiry from a brand name fragrance company where they found some imitation products on market. And they smell exactly the same, but the imitation is cheaper. So they want to find out if these two products are chemically identical or if there's any difference. Another thing they notice is that after you spray the imitation product on you, after a while, it doesn't smell as good as initially. So they wonder if it's because of some difference in the chemical constituents. One analysis we did for them is the TGA. TGA is a thermal analysis technique where you heat the samples and then you monitor the weight loss. So, for example, for fragrance, you have solvent like ethanol, which has a very low boiling point. So when you heat the samples, you will get a weight loss because the ethanol evaporates. And then you also have some other fragrance components, which has a relatively high boiling point, so they will evaporate later, which will give you a second or third weight loss step. What we observe here, as we can see, that both designer and the imitation products contain ethanols, and they both have weight loss at the first step. However, the weight loss of imitation product is higher than the designer product. It's an indication that there is more ethanol in the imitation product. So that's why it's cheaper, because they just spike their product with more ethanol. And another thing we notice is that the fragrance in the brand name has a higher content compared to the imitation. So after this, we did a DMC GCFID, which can give you detailed information on the chemical constituents of volatile compounds of these two products. And we compared the chemical profiles of these two compounds products, and we found the following observations. The first, compared to the imitation, the brand name fragrance has more expensive fragrance components in their product. And second, we found that there are some UVB absorbers, which is unique to this brand name product. They put this product into their they put this compound into their product to stabilize the fragrance. So the fragrance can stay longer on you after you spray it on you. And this explains why the imitation doesn't stay long and it doesn't smell as good after a while, because there's no stabilizer in their formula. So earlier I talked about the deformulation, where you can use it to look into the chemical constituents of a certain product and also compare different products. And another thing we can do here at Jordi Labs is the quality control testing. We do this type of test to help our customer monitor the reproducibilities of their products. We also help our customer to figure out the failure of their product if we see inconsistency of their products. For example, in the slides we show here are the two batches of soap samples we obtained, and we compare the GCMS and LCMS chromatograms between batches, and we can see that the two batches are pretty reproducible. It's a good indication that it's a good quality control over their product. One thing I want to mention is that sometimes we might see impurities, we might see inconsistency of one compound or several compounds in their product, and with the utilization of GCMS and LCMS, we can actually give you the identifications of the structure of their compounds, and it can help our customer tremendously to do the troubleshooting if they see some quality failure in the manufacturing process. Another quality control type of testing we can do is the purity test. This is actually a project we got from a bar soap, where they observed some crystal-looking particles on the surface of the product. When the customer used it, it was reported to have an uncomfortable sandpaper feeling when you rub it against you. So the purpose of this project is to find out the chemical components of these crystals, what they are. What we use here is FTIR to look into the bulk chemistry of these crystals, and we isolate the crystal from the soap, and we compare the FTIR spectra of these crystals with the soap product, and which is showing in blue and the red, and you can see the spectrum are not quite the same. It indicates that they are different things. So the crystal is something else, and then we compare our spectra with our library, which contains over 20,000 different compounds, and we found a good match, which is sodium gluconate. And as you can see that the sodium gluconate spectra matches perfectly to the spectra we obtained from the crystal-looking suspicious particles, both in the fingerprint region and the region over 3,000. So the sodium gluconate is actually added on purpose to the product to help for the leather, because it can sequester hot waters and make the product foam better. However, they encountered some solubility issues during manufacturing, and this is the cause of this insoluble sodium gluconate on the surface of the soap. And by a single FTIR analysis, we are able to help our customer for this problem they encountered. Next, let me move to a very special area for cosmetic analysis, that's pigment analysis, because pigment is very widely applied in the cosmetic product like makeups. The test articles we use here are two of my favorite eyeshadows. One is the brown matte eyeshadow, and the other is the glitter eyeshadow. What we did here is a SEM EDAX, where the EDAX analysis can give you the information on the composition of the product. Another powerful thing the EDAX can do is that you can pick the area of interest and then do the analysis on the certain area. So for the matte brown eyeshadows, we picked 3 representative areas on the product, and we did the SEM EDAX analysis. What we can see here is that these 3 areas has pretty similar elemental compositions. It's an indication that the product is homogeneous. And the elements we observed from these areas included silicon and titanium and oxygen and ferric ion. The silicon is widely applied in cosmetics product because they increase the fluidity of the product, and so the eyeshadow can glide well onto you and stay onto your skin. And titanium dioxide is also very widely applied in the industry. It can increase the opacity of the product. It's a good pigment, and it's also a sunscreening. Another thing we see is iron. I guess this is used to give you that brown color of this eyeshadow product. Next is the glitter eyeshadow. So again, we picked 3 representative areas and did the ASIM index on these areas. And what we observe is that these areas are actually not so identical. They're pretty different from each other. So this is the indication that the product is not homogeneous. Besides what we observed from the matte eyeshadow, where there's a silicon, where there's a soft titanium and oxygen, we also observed some heavy metals in the glitter eyeshadow, which is silver. So I guess they just apply silver to make the glitter particles, and so it will shine on you and make you look pretty. All right, last but not the least, the regulatory analysis. We sometimes get regulatory requests from the customer because certain compounds are actually banned to use in the cosmetic products. And we are helping our customer to make sure they're implying to the regulations. So for this particular project, the test articles are 3 complimentary shampoo samples I got from the hotel room. And we tested the presence of this coconut DEA in the products, because the coconut DEA is prohibited to use to produce cosmetic products. They are carcinogens, which has adverse effect on your body. And so for this project, we want to screen targeting on this compound to see if they're present in these shampoos. And it turned out, by using a target analysis by extracted ion chromatogram, we found out this compound is actually present in the blue shampoo showing earlier. So I guess afterwards, I just stay away from all the hotel shampoos I got. Another thing we did is the analysis on PAA. Those are primary aromatic amines. PAA are a group of compounds which again can cause cancer and can have some adverse effects on human, even at a very low level. And that's why in this particular project, we created standards containing different types of PAAs, and we use Q12 LCMS to detect the presence of these compounds. The reason we use Q12 LCMS is that it has a very low detection limit, and it can detect trace level of compounds at PPT, parts per trillion levels. What we did is that we prepared standards containing these PAAs at a level from 0.1 nanogram per ml to 1 nanogram per ml, and we generated standard curves. First, we want to see if we can detect these compounds at low level, and second, we want to see if we can generate a good standard curve. And as you can see here, we are able to detect all of these compounds and obtain a chromatogram in a relatively short period. It's less than 15 minutes. And another thing is that we can get very linear standard curves from each of these compounds. If I show you the R-square values for all the calibration curves we constructed for each of the standards, you can see they are all close to 1. So this is an indication that the method is very good and can give you accurate quantifications on each of these compounds. Okay, so comes to our summary. In today's talk, I covered different aspects of cosmetic analysis and how we can use the up-to-date techniques to help us solve different problems. We can use LCMS, GCMS, and other spectroscopy methods for deformulation. The deformulation can allow us to look into the chemical constituents of a product, and it can also allow us to compare products to products. We can also conduct quality testing and help our customer to monitor the reproducibilities from batches to batches. We can do purity test to troubleshooting why they observe a failure in a certain product. Another thing we can do is a pigment test. For example, for some cosmetic products like eyeshadows and makeups, these are very important to do. And next, it's the regulatory tests where we can analyze at trace level of the certain compounds which are inhibited to use for cosmetic products. So if you have further questions or you encountered any problems for your products, you can just visit our website, and our trained, very experienced chemists can answer your wide questions.